Hello, everyone. My name is Tom Giannasca, and welcome to the virtual presentation of my honors thesis work as part of the Commonwealth Honors College at UMass Amherst. For my honors thesis, I have been investigating how maternal PFOS and PFBS exposures impact ovarian transporter expression, driving alterations observed in maternal transfer of nutrients in embryos. In this presentation, I will introduce perfluoroalkyl substances, also known as PFAS, the use of zebrafish as an experimental model, explain the basic cellular background of my project, as well as show the method's results and conclusions. As I mentioned before, perfluoroalkyl substances are also known as PFAS, and they are compounds frequently utilized in industrial manufacturing, but they are mostly known for their use in firefighting foams, nonstick products, and textiles. And the reason these compounds are of interest is because they have shown to disrupt cellular function, have been found in significant concentrations in environmental media, such as drinking water. But more importantly, these compounds can enter and react with our physiological functions, as they are found in human urine and blood serum samples. So, the effects of two particular PFASs were investigated in this project, one being perfluorobutane sulfonic acid, or PFBS, and the other being perfluorooctane sulfonic acid, or PFOS. And these are their chemical structures, PFOS being a long chain PFAS and PFBS being a short chain PFAS. The difference between chain length is important because it is shown to affect the toxicokinetics and uptake by transporters, which is pertinent to this study. These compounds also have the potential to interact with our cellular function for a fairly long time, PFBS having a half-life of 44 days and PFOS for 4 to 5 years. Another important characteristic of these compounds is that they can induce oxidative stress in the cell and really disrupt the redox environment. This can be detrimental to physiological function of the cell because reactive oxygen species induced by PFAS can oxidize the active sites of proteins and disrupt their function. It is important to note that PFOS is a known endocrine disruptor and has mostly been phased out of industry. But PFBS has largely taken its place in industrial practices, making it a target of toxicological studies. At the Timmy lab here at UMass Amherst, we utilize a zebrafish model to investigate how PFAS can disrupt developmental function. And generally, zebrafish can be considered a great model for cellular investigations because their genome contains 70% similarity to our genome. Furthermore, their genome can also be easily manipulated to create transgenic lines that aid in investigations of cellular pathways. However, zebrafish are particularly good models for studying development because breeding can be easily induced and the embryos can be easily visualized and they develop very quickly externally. So the transgenic zebrafish line that was utilized in this study involves the NRF2A pathway. And cellularly, the NRF2A pathway induces the primary response to oxidative stress. So over to the left is the NRF2A pathway, and you can see here that KEAP1 ubiquitolates NRF2A for proteasomal degradation under normal conditions. And when there's oxidative stress in the cell, the NRF2A transcription factor is activated and localizes in the nucleus before binding other small proteins and eventually DNA to induce the expression of antioxidant proteins. However, there are other pathways that respond to oxidative stress, such as the PPAR pathway. So the NRF2A mutant zebrafish is a functional mutant that inhibits its ability to bind DNA. And the purpose of using this is to evaluate the role of oxidative stress in gene expression, but also identify potential genes that could be modulated by this NRF2A activation. So the goal of the study was to evaluate the gene expression of a few genes that may affect the nutrient loading of oocytes by the ovarian cell following PFBS and PFOS exposures. Two of these genes are apical and endothelial transporters, known as organic anion transporting polypeptides, 1D1 and 2B1, and are encoded by the genes SILCO1D1 and SILCO2B1 respectively. And these transporters normally function to deposit physiological hormones from the blood or the extracellular space into the ovarian cell. And it has been found that these transporters can also transport PFBS and PFOS directly into the cell. The other gene, LRP2A, encodes the protein megalin, and megalin is part of an endocytic receptor complex known as MERK that is thought to load the maturing oocyte with the proper nutrients for embryonic development. This is important to study because it has been previously shown that a suboptimal nutrient profile in developing embryos leads to later life metabolic dysfunction. So here's the predictive cellular model showing the ovarian cell with the genes of interest and the maturing oocyte. As you can see, OATP1D1 and 2B1 are capable of transporting PFAS into the cell and create oxidative stress. And the goal is to determine how oxidative stress and NRF2A affect the gene expression of these transporters. But also megalin, as changing levels of functional megalin may lead to this disruption of oocyte nutrient loading and subsequent later in life metabolic dysfunction. So how is this done? Well, maternal zebrafish were exposed to 3 different concentrations of either PFBS or PFOS for 1 week, and then the ovary tissue was collected at either the first breeding event or the 5th breeding event. And this was repeated a few times with both NRF2A wild types and NRF2A mutants. These concentrations of PFOS and PFBS are higher than those found in environmental media, but higher doses were predicted to elucidate measurable cellular response to these compounds. Following tissue collection, RNA from each sample was isolated and complementary DNA was synthesized. The gene expression of 5 genes in each sample was analyzed via quantitative real-time polymerase chain reaction. The 5 genes are beta-actin and beta-2 microglobulin as housekeeping genes, as well as LRP2A, SILCO1D1, and SILCO2B1. And we use these housekeeping genes to calculate the relative gene expressions of our genes of interest because the expression of these housekeeping genes should not change based on genotype or experimental treatments. New primers were designed for SILCO1D1 and SILCO2B1, and the annealing temperatures were optimized using a specialized thermal gradient PCR technique. And the PCR products were confirmed using agarose gel electrophoresis. So here we have the PCR gene expression data from the PFBS exposed fish. And as you can see, the only difference here is the decreasing trend in the SILCO2B1 gene across genotypes and treatment groups, but no significant changes in either the LRP2A or SILCO1D1 genes. And here is the expression data from the PFOS exposed fish. We can see that the SILCO2B1 gene did not express differently than the controls, but the other genes did, suggesting this gene may be more susceptible to PFBS than PFOS. We have a slight decrease in expression in the LRP2A gene in the smallest treatment group, in the wild-type fish, but not the mutant fish. And the SILCO1D1 gene expression increased in the mutant fish, but not the wild-type fish. And here are these results just reiterated. So what does this mean as far as investigating the mechanisms of both oxidative stress and NRF2A activation? Well, because SILCO2B1 expression decreased in both genotypes, both the NRF2A and other antioxidant response pathways may modulate its expression. Similarly, SILCO1D1 increasing expression only in the mutants where the NRF2A transcription factor is ineffective applies another pathway may be at play, like PPAR. Because LRP2A decreased expression in the wild types and not the mutants may mean that this gene is modulated by the NRF2A pathway directly. Changing the expression of these genes may have some cellular implications. Decreasing SILCO2B1 may prevent the proper amounts of hormones from entering the cell, and this may affect other cellular properties, especially in the ovary cell. Increasing SILCO1D1 may lead to even more PFAS entering the cell and causing even more damage through oxidative stress. And finally, decreasing LRP2A may result in a suboptimal nutrient profile that may impede proper embryonic development. What may be happening here is that the NRF2A is activated following exposure to oxidative stress, and this purposefully downregulates LRP2A so that the maturing oocyte is not inadvertently loaded with toxicants. However, as I said before, this would also decrease the amount of nutrients loaded into the cell. So how does this relate to human health? Well, this decrease in LRP2A at the lowest concentration follows a trend of many PFAS where cellular response can be measured at lower and lower doses. And this suggests that the lower concentrations found in environmental media may have an exaggerated effect in humans than even higher doses. And here are these discussion points reiterated. And overall, this work and other research on PFBS and PFOS are important because they show that these compounds do induce adverse effects in cellular function and may contribute to the development of later in life metabolic diseases like diabetes or obesity. And to further investigate the possible effects of PFAS exposures, PPAR could be measured in the NRF2A mutants in order to elucidate the effects of antioxidant pathways and the preferences for one pathway over another. Nutrient panels could be utilized, or more specifically an amino acid panel, to show that these progeny are in fact affected by the mother being exposed to these PFASs. And if the progeny do survive the mother being exposed to the PFASs, they could be subjected to later in life metabolic evaluations such as glucose challenges. This thesis project was funded by the Commonwealth Honors College at UMass Amherst and the NIH. And I really want to thank Dr. Timmy Larragee, Dr. Annunziato, Marjorie, Sarah, and the rest of the TL lab for all their help and expertise that allowed me to complete this project. I hope you enjoyed listening to my honors thesis work, and thank you for watching.